Today we are joined by Andrey Kovalevsky, a structural biologist and biochemist at the Oak Ridge National Laboratory. And we're going to be exploring how they used MedChem Tool in Nanome to develop a new potential drug against the main protease of the SARS-CoV-2. Hi, Andrey. Thanks for joining.
Hi, Happy New Year, everybody. We have started with this molecule shown here in the X-ray structure. It's a non-covalent inhibitor that was discovered through our collaboration with Argonne National Lab, who did the high throughput computational screening of non-covalent compounds. This molecule has three parts: the P1 group, which we call uracil, the linker, the piperazine ring, and P2 group, which is here at dichlorobenzene. They fit into the two sites in the main protease. Uracil fits in the S1 site. P2 group fits in the S2 site, and the linker is actually sitting above the catalytic dyad, which consists of cysteine 145 and histidine 41. With neutron diffraction, we were able to determine the protonation states as shown here for this molecule. This nitrogen turned out to be protonated. So this hydrogen here is actually experimentally determined hydrogen. This nitrogen is positively charged. Therefore, we also see the hydrogen atoms on these nitrogens as well, and we can directly visualize hydrogen bonding that the carbonyl groups here can make, and this one as well with the protease. So we started with this molecule, and we decided to use Nanome and the MedChem Tool to perform structure activity relationship study, which means changing chemically this molecule into adding or altering substituents to change its hydrogen bonding properties, electrostatic properties, or electronegativity properties of different groups, and see how they fit in the pockets. So we can start with going into the S2 pocket here.
Right.
Yep. And showing what we actually did. It the molecule carves out a really nice deep S2 pocket shown here. As we see here, it's covered from the top with a glutamine 189 residue. So now we can see what we can change. We can basically alter substituents. These chlorines can be altered, and a new substituent can be added also here, for example, on this meta carbon. So if we remove the surface, we can see some of the residues that are surrounding this molecule that are important for interactions. For example, methionine 49 right here is an important residue that interacts with many inhibitors. It also is known to be very flexible, so it can change the conformation, and we can add substituents that would not clash too much with the protease. There's also cysteine 44 down there that provides some properties to interact with substituents. So, for example, on this carbon atom, we added a chlorine. So we can go to MedChem Tool and add the chlorine atom there.
Yeah, that's great. Yeah.
One of the reasons we did that instead of adding something very big is because even though methionine can move away there's still not much space to add very bulky substituents. So chlorine was one of the interesting ones to add. So if we can add it,
we can place a chlorine over here, click on the controller, and boom, place a chlorine here. See how the molecule auto minimizes a little bit?
Yes, and you can now clearly see that the chlorine is not clashing with methionine. It may need to push it away a little bit. It is close to cysteine 44 and may be interacting pretty well with it. It turns out that when we did this, the compound was a better inhibitor than the initial one. Means that the chlorine has the right electrostatic properties, right electronegativity properties, and it does interact with cysteine 44 favorably to increase binding of this new compound to the main protease. We also changed the substituents to replace this other two chlorine atoms that were from the starting molecule. And the reason we did that, for example, for this para-chlorine right there, so if we remove the surface, we can clearly see why we did this. There is a residue nearby, which is tyrosine 54. And there it is, tyrosine 54. It's the phenolic oxygen is supposedly a good hydrogen bonding donor or acceptor. So we thought that by substituting this chlorine with, say, CH2OH group or aldehyde group, we can engage this hydrogen bonding. We obviously also were interested in seeing whether this can happen. This OH group is hydrogen bonded already to a different main chain carbonyl. So the question was, can we use this oxygen as a hydrogen bonding acceptor by adding CH2OH or aldehyde to improve binding? For structural activity relationship, it's important to know what you can and cannot do by substituting different groups. It turned out to be if we can actually build CH2OH group here instead of chlorine.
Sure. Let's delete this measurement. Okay. We're going to place it on the para chlorine or
here? Yes.
This one here.
Yeah. Instead of chlorine, it should be carbon here.
Oh, sure. So let's delete the chlorine first, so we can just get rid of this one.
Okay.
And now I am going to build that group.
CH2OH group.
Yeah, there we go.
Yes.
I think that makes sense. Boom, there you go. Yes. Yeah, we got some hydrogen bonds as well.
Excellent. So this is exactly what we thought is going to happen. The main protease doesn't actually like the CH2OH. Even though we can build it, we can see that perhaps this would help. It actually doesn't help, and it could be because of electrostatic interactions, because of OH group maybe not having the right geometry to actually connect hydrogen bond to this tyrosine. Obviously, when we don't have clear knowledge about where hydrogen atoms are, it's hard to theorize what will happen. But that was an important observation when we did this, and it gave us an idea that such bulkier, more electronegative substituents in the para position of this benzene were not going to be good for binding.
I see.
We also changed this chlorine into various substituents, and it turned out that the chlorine actually is the best one here, but we were able to change this chlorine to fluorine atom, to CF3 groups as well, and the electronegative groups were not good for this position, for example.
I see.
That
Yeah, so we're gonna go back to your molecule anyway.
yes,
And yeah, here too, I'm gonna delete this group and I'm gonna build again the chlorine.
chlorine.
Yeah. There you go. So this is the actual compound that your laboratory has developed, right?
Yes, and
Nice.
what's important also, as we saw in the very beginning, this nitrogen is very important. Its protonation that makes it positively charged is beneficial for binding. If we replace this nitrogen with carbon, which is basically has the same geometry, is just not charged. If the NH plus is changed into CH plus, the binding is not as good.
Right.
Which we also learned, which could be because the catalytic dyad here, cysteine histidine catalytic dyad, is neutral when the non-covalent inhibitor binds. Cysteine is protonated, it's thiol. Histidine is not doubly protonated, has only one hydrogen, so it's neutral in charge. This plus charge on this nitrogen is important for binding, it seems. If we go quickly to the S1 side, here, this hydrogen bond between the carbonyl of the uracil group is a crucial hydrogen bond to histidine 163 for binding. So what we did, we thought that we can try to disrupt a little bit this hydrogen bond to see if it's indeed a very important one. So to do that, we changed this specific double bond here on the uracil into a single bond. So this carbonyl, this carbon atom and this carbon atom become sp3 hybridized and become tetrahedral, which actually changes the geometry of this ring. So the distribution of double bonds should be different here. But what's important is when these two carbon atoms are not connected by a double bond, when it's a single bond and this carbon is tetrahedral, the binding of the molecule, this group, actually disrupts this hydrogen bond to histidine 163 enough to make it a worse inhibitor. So by doing that in Nanome, by looking at that, we figured out that this hydrogen bond for the inhibitors is one of the most crucial hydrogen bonds for drug design.
Interesting. Oh. Okay, yeah, got rid of the hydrogen bonds in there. Nice. So yeah, we have this beautiful molecule. It just happens to be way more able to bind according to the IC50 values, right? According to your
yes,
publication.
It's several times better binder than the initial molecule, and it becomes a very good starting point. We can call it a lead compound to design further substituents that may improve the binding and make this compound antivirally potent. So these studies are in progress right now.
Are you doing some clinical trials soon, or we're not there yet?
We're not there yet. We need a compound that would be a very good antiviral. It should stop virus from replicating, and only from that stage we can go into testing it in vivo, in perhaps in animal models. Right now, this study specifically looked at structure-activity relationship of the two pockets and the linker above the catalytic diet to figure out what is important for binding.
Right.
It turned out to produce a better molecule, which is very good and gives us ideas how to further improve. But most importantly, we figured out what the electrostatic, hydrophobic, and steric determinants are for the binding to these two pockets in the main protease.
Right. Yeah, it's fantastic to see how your team at Oak Ridge National Laboratory has been carrying out this project with the cross-disciplinary scientists, right, and many different departments are working together with different techniques.
Yes.
Actually, both in silico, experimental. It's just amazing
Yes.
how you've come up with this, and also using virtual reality to gain all these insights in terms of the three-dimensionality, and also making designs actually using the MedChem tool in Nanome, right?
Yes, absolutely. We most importantly, we were able to visualize the way you can't visualize in any other program this molecule and the interactions, and then go from there by building these molecules in silica and then testing them in the in solution, growing crystals, producing X-ray and neutron structures, doing molecular dynamic simulations, and also we looked at thermodynamics of binding using isothermal titration calorimetry, which is a very good tool to understand, to kind of connect the structural features to the binding energies.
Nice. Well, thank you very much, Andrey, for joining us today and explaining how you and your team were able to develop this potential drug for fighting COVID-19. We're happy at Nanome, of course, that you were able to use our tool to visualize and design these drugs along with other tools. And yeah, thanks again. Thank you, everyone. And we'll see you next time.